To conclude our discussion of gene expression and transcription and translation, we have a few topics that will sort of round out our perspective on the entire process. That includes talking about the way that introns are spliced out of the initially synthesized eukaryotic mRNAs, the transport of mRNA from the nucleus to the cytoplasm. We'll talk about how protein folding can be assisted by chaperones. We'll talk about how proteins are degraded. We'll talk about a little bit about the proteasome, which is sort of it is the protein complex that degrades the cellular protein, sort of the anti-partner of the ribosome. And then we'll just talk a little bit about how antibacterial drugs target the ribosome as a way of trying to cause bacterial cell death. Here I'd like to just briefly show some of the data that was used for the discovery of introns. As you can see in this case, where you're going to hybridize some mRNA to DNA to find out where the gene is. If there are no introns, the mRNA sequence will be an exact copy of the genomic DNA. However, if the mRNA have introns spliced out, part of the mRNA will hybridize to the part of the genomic DNA for exon 1. Genomic DNA corresponding to the intron will not hybridize to anything in the mature mRNA, but then this other part of the mRNA will hybridize to part of the genomic DNA for exon 2. And these kinds of structures are observed, like for instance here, and that was how the existence of introns was deduced. Just like the sequences that help proteins recognize where transcription should start and stop, there are sequences that help other proteins and RNA molecules recognize that these are exon-intron boundaries and that there should be a splicing event here. Now, not all of exons are used, and not all splices are simple splices with the contiguous regions being spliced together. Sometimes you might skip exon 2 and go to exon 3. The way that is determined by the cell, that is not entirely clear as far as I'm aware. And there is an 8 base pair sequence at the 5' end and a 12 base pair sequence at the 3' end that indicates where the splicing should occur, and here is where the excised intron would have been. Now in the sequences here, in addition to A, C, G, and U, there's some other characters here, other letters. That's Y is for pyrimidine, R is for purine, meaning it could be either A or G. Pyrimidine means it could be either U or C. In the center here you see some of the sequences that are involved, in particular the A, which does some of the chemistry of the splicing reaction, which is also done by RNA molecules. Splicing is performed by a protein and RNA complex called the spliceosome. It's a very large, complex machine, and an example of the part of the RNA doing some of the chemistry involved with the splicing of exons, and that is that base pair A that was in red in the previous slide, attacking the upstream exon-intron boundary, forming this lariat structure, which then is cleaved out. The two exon ends are joined, and now you have a spliced boundary region of your mRNA. Now, this is one of the first examples of RNA having enzymatic activity, and the fact that RNA is a molecule that can both code for information and perform lots of biochemistry has led some people to consider that speculate on the possibility of RNA being one of the very primordial molecules present at the origin of life, and that's as good a hypothesis as any right now. One of the aspects of the differences between eukaryotic and prokaryotic RNA that we pointed out was a PolyA tail at the end of 3' end of eukaryotic mRNAs. Once the RNA stop sequence, or the stop sequence for RNA polymerase has been passed, the RNA polymerase will dissociate from the DNA, dissociate the RNA from the complex, and an enzyme called PolyA polymerase, a polymerase which does not need a template, will just start adding AAAAA to the end of a synthesized mRNA. This PolyA run will be covered with PolyA binding protein, and it will help be involved in the transport of mRNA out of the nucleus. The fact that all eukaryotic genes, mRNAs, end in PolyA, those that are protein coding, has been tremendously useful because it allows scientists to take a poly-T primer, which will bind to all the RNAs and synthesize them for making molecules called cDNA, and we'll see that in future lectures. Once synthesized and processed in the nucleus, mRNAs need to be transported out into the cytoplasm. In the nucleus, they're bound by the cap binding protein, the PolyA binding proteins, and a complex called the exon junction complex. Movement inside the nucleus is partially aided by diffusion. Once it goes through the nuclear pore, large openings in the double membrane that separates the nucleus from the cytosol, proteins will bind to the cap region and the mRNAs and help transport them to ribosomes at the sites where they are synthesized into, or read for protein synthesis. We saw earlier in the course that proteins could be unfolded with urea, full-length protein sequences could be unfolded with urea and then refold on their own. Now, as they're being synthesized, the amino end of a protein is exposed prior to completion of the entire polypeptide chain. So proteins can start to fold in domains before the entire sequence is even available. This shows you that kinetic factors are potentially can play an important role in the final folded structure. If this portion of the protein starts to lock into a particular configuration, once full-length protein is present, it may not be able to unfold what's already been performed, and this is one of the reasons why we're not certain that most proteins fold to a global thermodynamic minimum, because these proteins can get kinetically trapped into a local minimum by the process where part of the protein starts to fold before the rest is present. Sometimes proteins don't get folded correctly due to random events. Other proteins were never intended to fold entirely by themselves and require some help. A class of proteins called chaperones play large roles in the folding of many proteins and the correction of incorrectly folded proteins. When a protein is folded incorrectly, it will interact with a chaperone, which will then attempt to refold it, or it can also send it for degradation to the proteasome, which we'll see. This can happen a couple times, and every time that it goes to the chaperone, there is a chance that it will be shuttled, marked for destruction. However, it's correctly folded, it will no longer interact with the chaperone, and it can diffuse away to perform its function. I think it's pretty amazing that this system can operate in this way, and you might think for a few seconds about how could the chaperone possibly know if the protein is folded correctly or not. Please pause, and we'll resume on the next slide. Okay, here's how it knows. Proteins that are folded correctly have hydrophobic cores and hydrophilic surfaces. If a protein is incorrectly folded, there's a good chance that a hydrophobic patch that is meant for the interior of the protein will be exposed on the surface. The chaperones have hydrophobic binding regions which can interact with these incorrectly folded proteins. Neither of the hydrophobic regions want to be exposed to the solvent, so that they will be lower in energy if they are interacting with each other. This is a similar situation to what we saw with the globin protein in sickle cell anemia. Now here, with the help of ATP binding and hydrolysis, the protein will be drawn into the interior. A cap will form, and now it is sort of isolated from other external influences, and it will attempt to refold it. It then can be released, and if it's correctly folded, it will diffuse away because it no longer has an exposed hydrophobic region to interact with the chaperone. But if it is incorrectly folded, it may interact again and go back. This entire process of a protein entering the chaperone, refolding, and being released occurs in less than a second. And how the chaperone manages to unfold and let a protein refold, I'm not fully aware. I suspect that it is not completely understood. The two cavities of the chaperone are essentially identical, though only one of the two cavities is active at any particular time. Here we see the proteasome. This is the structure that targets nearly all of the protein for degradation. It is also a very abundant protein. It can be also 1 to 2% of the cell's mass. If you think about it, every protein that gets made by the ribosome can be degraded by the proteasome, and so that it has a very important role in the overall maintenance of protein level. In this sort of flow chart, it's mostly saying protein aggregate is destined for the proteasome, but any protein where the cell no longer needs it, it marks it for destruction and shuttles it to the proteasome. Ubiquitin is a small protein sequence that gets covalently attached to an existing protein. Now, a single ubiquitin, one of these red subunits, can be a signaling indication. There's a lot of functions for a single ubiquitin on a protein. However, if there are multiple ubiquitin units on a protein, this protein is marked for destruction. And that occurs because these ubiquitin units interact with the proteasome cap proteins, and again, it's just thermodynamics. If they encounter, they will bind. If they bind, it will be threaded into the center of the barrel. I'll go back. The blue subunits, and there are 3 different types of proteases inside this chamber that will digest the proteins into single amino acids, which then can be reused by the cell for additional protein synthesis. It's still a very active area of research in terms of figuring out how the cell knows which proteins should be tagged for destruction if they're properly folded and simply no longer needed for utility. But the basis of most of this is 3 types of proteins that form the ubiquitin ligase machinery. Your human genome has a few E1 units, maybe a dozen or so E2 units, and maybe around 6 or 7 dozen E3 units. And so combinatorially, you can have potentially many different E1, E2, E3 complexes, and each of them has the potential of targeting different proteins. We are, however, a long way away from understanding which complexes target what proteins for what reason. In any case, the E2 unit will transfer a ubiquitin to a protein that it's going to mark for destruction. Additional ubiquitin subunits will be added to the chain, and now this protein will soon be degraded. Here we see the bacterial ribosome and highlight many of the antibiotics that target the ribosome in an effort to kill bacteria. Bacterial ribosomes and eukaryotic ribosomes are fairly different, so that when you take tetracycline or erythromycin, they will leave your ribosomes alone, but they will bind to the bacterial ribosomes. You may know that when a drug is given to a bacteria, it will try to mutate so that the drug will not bind and so that it can escape cell death. The ribosome is such a complex machinery which has to be so careful in recognizing with great fidelity each of the 61 different tRNA molecules that it doesn't have a lot of room to maneuver in terms of mutations to avoid the being bound by the drugs and still function properly in terms of making protein synthesis. However, as you know, there's resistance to nearly every antibiotic that we have, and so the bacteria have found ways, but it's still an attractive target because of the many constraints on the design of the complex. I include this slide as a review of all the things we've talked about with transcription, processing of mRNAs, translation, protein folding, protein degradation. This slide is a good review for you because it captures a lot of information as you prepare for the first midterm. Thanks.